So today, let's talk a little bit about nutrition and exercise and how it affects our genetics. So things we'll be going over today. How do we interpret signals from our environment? How do cells talk to each other? We've talked a little bit about this in the past. What is epigenetics? This will be a new concept today of how our genetics can actually change and respond. And then the role of diet and exercise in epigenetics. So really today's theme is that we're not truly at the mercy of our genetics. We can affect positive change through a healthy diet and exercise. All right. So when people say that, I've inherited these features of mine and there's nothing I can do about it. Let's just use overweight, for an example, not to pick on overweight people. Yes, you can. And you can actually change your own genetics through food, diet, and good, healthy habits. And these genetic changes that we call epigenetics, that we'll talk a little bit about, can be transmitted to one's offspring, your genetic heritage. So you can actually, so if you look here, we've talked about vitamins, amino acids, fats. We've talked about metabolism in the past. How glucose is metabolized, how we make energy from it. We've talked a little bit about cell signaling, or the cytokines that are released from our cells. And remember, we've talked about the microbiome, the bugs inside of us, and how they participate with us, how we're symbiotic, how we have we coexist with each other. We've talked a little bit about gene expression, which is the nucleus of the cell, but not a lot on how we can actually change our own genes. So that's kind of what we'll talk a little bit about today. So just a little bit about myself for those of you who are new to the podcast. What is a surgeon bodybuilder? That's kind of me. So I've been playing, I'm 59 years old now, so I'm pushing 60 real quick. I've been playing sports since the age of 8. I played ball. I was a varsity letterman in football and wrestling. I continued weight training throughout college, grad school, med school, general surgery training, and vascular surgery fellowship. That's 17 years after high school. Obviously, I was focused on becoming a surgeon at that point, but I was able to continue weight training throughout that. I finished my fellowship in 98, so I've been in practice for 25 years. I'm a full professor at University of Miami in the Department of Surgery, so I teach in addition to my own practice. So it's been a total of 44 years of weight training. I've been doing competitive bodybuilding for 20 years. I've done 17 shows, meaning I've gone through the bulk and cut process 17 times. So those of you know a little bit about these diets, they're pretty strict, and you really learn how to tune in on the macronutrients and micronutrients, proteins, carbs, and fats. And we've talked about that in the past, how it gets translated energy, and how you can change your body composition. And that's what we'll start talking more about in the future. But what I'm trying to do here is lay a foundation of knowledge so folks can truly see why diet and exercise are important. We've heard it our entire lives, healthy diet and exercise is the key to health, but people don't do it. And maybe it's because they don't understand why. So that's what I'm trying to do here with the podcast. So I've been weight training for 44 years. I'm also a gym owner for the past 7 years. Been doing karate for 27 years, do private lessons in the mornings. Still do that. And my best achievement is, and it's sad you have to say this, married to the same wife for 30 years. So my wife and I have been married for 30 years. We have 4 great kids. They've all made it through college. Nobody's on drugs. So that's our crown achievement, mostly because they had a good mother, or have a good mother. All right, enough about me. That's just kind of a backdrop as why I'm into this sort of thing and have adopted it into the practice. MiamiBain.com, MidtownBody.com. So we've got a team of people that the last few years have been getting more into this, trying to help people with diet and exercise, what we call wellness part of the practice, or some people call it functional medicine. I just call it common sense, trying to get people to eat better, to get off all these medications we see them on, lose some weight, and get healthy. That's really all we're trying to do here. And hopefully by educating you, and I'm trying to make this technical enough where, medical or professional scientific people make sense to them, but even just normal people, just to not overwhelm you with all the mechanisms, but give you just an understanding of what we're talking about. So back to a little basics. Again, prokaryotes are one-cell organisms, eukaryotes are multi-cell organisms. So we're mammals, we're eukaryotes. And really what that means is our cells, this is one cell, have a nucleus and have cytoplasm. And within the cytoplasm, there's things like the endoplasmic reticulum, where protein synthesis, really where the DNA message gets transcribed and then translated into proteins in the endoplasmic reticulum. Lysosomes are kind of disposal units of just one function, and we'll talk about this a little more when we talk about mTOR. But multiple things go on in the cell. Last time we talked a lot about the mitochondria, the powerhouse of the cell. And today is more about the genetic material inside of the nucleus, which is DNA. Most people know this. We're going to try and just establish a little more about DNA and how the blueprints of our person gets expressed. Cytoplasm is where anaerobic glycolysis occurs, the first part of metabolism, and mitochondria, where the oxygen gets used for aerobic respiration. So just a quick review on that, what we call the macros: fats, carbohydrates, and proteins. This is our foodstuffs that come into the body. They get then broken down to their basic structures, which are fats are made of fatty acids and glycerol, carbohydrates to glucose, and proteins to amino acids. So these are the building blocks of the macronutrients. We bring them into our body, break them down from foodstuffs or complex meals into these building blocks, and then absorb them and then rebuild them in our bodies for whatever needs we have. So glycolysis is what happens when we start breaking down sugar without oxygen, and in the presence of oxygen, glucose, which is a 6-carbon molecule, gets broken down into 2 3-carbon molecules called pyruvate. It spins off a CO2, and then acetyl-CoA, and we'll talk about this at the end, is what really goes into the mitochondria, into the Krebs cycle that we've talked about a lot. And this is where ATP is made, our energy. This is the molecule that gets split off into ADP plus phosphate, and energy is released. Our mitochondria have the electron transport chain, which is the battery. Electrons, or electricity, is literally conducted down our inner membrane of the mitochondria to produce ATP using a proton pump. So we've talked about this in previous videos, and you can, if you want to review this, you can do that in previous videos. This just emphasizes how we use food and how we make energy out of it. Okay. Part of the other thing of the Krebs cycle, or the TCA cycle, is that intermediates are being siphoned off for biosynthetic purposes. The cycle has to be replenished or else it will cease. So I'm just showing this. This is a 6-carbon molecule called glucose. You can see that oxygen and hydrogen are attached. There's bonds, but this is a 6-carbon skeleton. And the point is that the carbon skeleton goes into this Krebs cycle, and a 6-carbon glucose goes into then as a 5-carbon alpha ketoglutarate, 4-carbon oxaloacetate. So it spins through here. These carbon skeletons are repurposed. So not only is it used for energy, but things will spin off of here to make things like the nucleotides of DNA, to make things like building blocks for proteins, etc. Okay, very busy. Uh, the mitochondria is a very busy organelle with respect to energy production and biosynthesis. So how hormones and receptors, what are they? And this is the nucleus and this is the DNA. So we've talked about cells talking to each other. So hormones are something that a cell could produce to talk to another cell, either nearby or far away. So for example, the pituitary gland in the brain produces hormones or peptides signaling molecules that can, for example, have an effect down on the gonads, far away from the brain, down the gonads. The pituitary is talking to the gonads to make testosterone, or the ovaries making estrogen, for example. High testosterone will feed back to the pituitary and say, there's too much testosterone, let's turn down the production, or vice versa. So how does all that happen? Well, hormones get into the nucleus and attach to a receptor, and you'll see a little bit more about this, but these receptors then activate a whole cascade of intermediaries or molecules that then can go into the DNA, and these are the directions of what the cell produces on our DNA, the genetic information. So by binding to the DNA, different messages can be produced. DNA gets copied into messenger RNA. Messenger RNA goes out of the nucleus to the ribosome to produce a new protein. So this is the process of transcription, which is DNA to RNA, and translation, RNA to protein, RNA to transfer RNA to protein. Okay, these are amino acids being linked up into a protein. This is a ribosome. All right, so what I'm trying to emphasize in this talk is how our body can respond to environment or nutrition or exercise, and changes can be made to our DNA to affect change or the production of new proteins or new molecules that will respond to us as an organism to either create a positive change or a negative change. And these changes can be inherited to our offspring. They can be permanent or they can be temporary. So this gets us to epigenetics. Many of you have heard of this. What is epigenetics? Epigenetics alters gene expression without altering the DNA sequence. It's heritable or inherited and reversible, as we've talked. And much of this is done through something called methyl donating units, or methyl donating nutrients in the diet. Methylation and acetylation, or acetyl groups, is another one. So what does all this mean? So most people know we have chromosomes, 46 chromosomes. Chromosomes are DNA wrapped up around histones. Chromatin is when it gets thickly wound around these histone proteins. And as we start unwinding things in the cartoon, just to show you what we're talking about here, DNA is that double-stranded double helix discovered by Watson and Crick that most people know about. So what people don't know much about, or in the lay public, is that DNA wraps around these histones. There's 4 histones here. And the gene can be accessible or non-accessible by how tightly wound this stuff is, like a yarn, wound up or loosened up. So by methylating or acetylating, one can loosen up the yarn or loosen up the DNA off the histone to make a gene more accessible for transcription, which then will go on to translation and production of protein, or it can be wound tight and inaccessible. So this process of epigenetics or methylation, acetylation can tighten up or loosen up the DNA so genes can be exposed to be read or non-read, so active or silent. That's what epigenetics is. Okay. Histone modification, the binding of epigenetic factors to histone tails alters the extent of DNA wrapped around. DNA methylation, we talked about, histones are protein which DNA wraps around. And it's felt that, cancer, autoimmune diseases, a lot of these have epigenetic factors that turn on or off these inflammatory type disorders. These types of things can change the epigenetics, like aging and diet, drugs, environment, poisons, exercise. All these things can have an effect to change epigenetics. So these things can cause a methylation or demethylation of our DNA. So different genes are transcribed either silent or active. That's all that's saying. And these is just to show you some peer-reviewed literature of how diet, the role of diet and epigenetics. So epigenetic mechanisms are heritable and reversible. Nutrients can modify physiologic processes through epigenetic mechanisms. Okay, this is what we've just said. It's just showing that literature supporting what we just said. Same with physical activity in the prevention of human diseases, role of epigenetic modifications. It's well accepted that physical activity and exercise can modulate gene expression through epigenetic alterations. Okay, just some literature. There's a lot of it out there to support what we're talking about here in the talk. This one's gotten a lot of attention and very busy slide. And I'm not trying to show this to impress you. I'm trying to show this to impress upon you the number of steps or reactions that occur for expression of mTOR. And just, so I've been studying this for 40 years since I was in college, and none of this information was available back then. So these researchers, these PhDs in these labs, these people are wizards, how they figure all this stuff out. But what I want to talk about here is that mTOR integrates nutrient availability and growth factor signaling. mTOR is kind of the growth, think of it as the conductor of the orchestra regulating growth. And the next slide, I'm going to show you the conductor of the orchestra regulating breakdown, which is ATP kinase. So mTOR, mammalian target of rapamycin complex 1. Okay, mammalian target of rapamycin complex 1. That's what mTOR is. mTOR changes activity in response to available amino acids, oxygen, growth factors, as well as decreases in energetic status of the cell. mTOR activation promotes anabolic pathways involved in protein synthesis, lipid synthesis, and can stimulate glycolysis. And so what this is saying is a growth factor. Insulin would be a classic one. There's amino acids particular to mTOR. Leucine is the big one. So let's just, everyday example. You take in a protein meal, and we've talked about this before. The protein needs to be broken down to amino acids. One amino acid, particularly called leucine, is the branched chain amino acid. It takes about 30 grams of leucine, or 30 grams of protein, which will house about 3 grams of leucine. 3 grams of leucine seems to be the activator, the on-off switch for mTOR, or just one of them. So just by eating enough leucine in protein-rich food, you can turn on mTOR and turn on growth. Okay, so what are some of the things mTOR regulates? mTOR has a positive effect on protein synthesis. Bodybuilders are interested in mTOR because they're trying to build muscle. Muscle is protein, actin, myosin. So by activating mTOR, we want to make bigger muscles. We need to eat protein, specifically leucine. Lipid synthesis are fats. Inhibits liposome biogenesis. It inhibits autophagy. Autophagy is a catabolic process we'll talk about next, where breakdown products need to be disposed of, or even bad cells need to be disposed of. We have mechanisms to get rid of waste products and bad cells called autophagy. mTOR obviously regulates energy metabolism. If energy is low, we need to make ATP. In order to make ATP, we need to turn on glycolysis and mitochondrial metabolism, which we said was oxidative phosphorylation, the creation of ATP. Okay, so I'm just kind of bringing all this stuff together into more mechanisms so you can understand why exercise and nutrition are important for health. So this is the growth signaling pathway regulated by mTOR. AMP kinase you can think of as the opposite. So where mTOR positively regulates anabolic or growth processes when nutrients are available, AMP kinase responds to increases in AMP. So remember, ATP gets broken down into AMP. So there's a lot of AMP around. It means that there's no ATP around, meaning things need to then change to restore or to turn into a catabolic processes to restore homeostasis or get us back to baseline. So AMP kinase, again, this is a conductor of the orchestra for catabolism. It inhibits fatty acid synthesis, the opposite of mTOR, inhibits sterol synthesis or lipid synthesis, opposite of mTOR, inhibits transcription of fat-producing genes. So these are the things that it inhibits. Down here, the things that stimulates, autophagy. So again, getting rid of broken products. Mitochondrial biogenesis, we need to make ATP. So it stimulates that. Fatty acid oxidation, I'm not sure why, because fatty acid oxidation produces ATP. So I'm not sure why this one's inhibited. Glucose uptake, again, you bring in glucose, break it down, and that's what goes into the anaerobic glycolysis, oxidative phosphorylation to make ATP. So these are the two big players, mTOR and AMP kinase. We're just showing how the steps involved into changing whether we need to get into a growth phase or a breakdown phase, and the fact that this sort of stuff all happens through gene transcription, getting the information from the nucleus to the outside of the cells. So these are, and we're just going to give a couple more examples. Oxygen also regulates transcriptional networks, which makes sense. So this is a normal oxygen, and when we're in a hypoxic situation with lack of oxygen, at the cell level, oxygen levels starting at 5% begin to noticeably activate HIFs, or hypoxia-inducible transcription factors. So these are factors that go to the nucleus to tell the DNA, listen, we need to turn on some genes here to reduce metabolism, for example. We need oxygen for oxidative phosphorylation to make the electron transport chain work, to make ATP. If we said in previous lectures, well, there's no oxygen around, it would make sense that the cell needs to start thinking about slowing down metabolism. So hypoxia is a molecule, lack of oxygen stimulates receptors, which then go into the cytoplasm, go into the nucleus to affect the DNA, to affect our genetic information, to tell the cell, hey, we need to slow down metabolism. Glucose is another one. We've talked about glucose a lot in previous lectures. Cells have a mechanism to monitor glucose levels. Of course, insulin is the main hormone that regulates insulin, but it does this by, again, talking to the nucleus. So glucose, we know that insulin causes expression of the GLUT4 receptor on the cell membrane to allow glucose into the cell. Then a lot of things occur inside of the cell that we have not talked about in previous lectures. That's why we're mentioning them here. But things like Mondo A and CHR, and these are just names that the researchers attach, but these are just intermediates in the process from glucose to telling the cell that, listen, we need to change our expression of proteins to deal with this glucose in the cell. And how's that happen? Through these CHREB phosphate and Mondo A. These are the guys that go to the DNA and act as promoter sequences to turn a gene on to be expressed. And that means that a particular protein will be made in response to glucose. That's all this is saying. And all I'm trying to say is that our environment, our nutrition helps affect our genetic information and the things that are read or expressed by our bodies. So genetics are not permanent. What we inherit is not permanent. I think this is one of the last slides. Acetyl-CoA, we've talked about a lot because remember, glucose gets broken down into pyruvate. Pyruvate then gets, as it throws off a carbon, acetyl-CoA is the key player that goes into the Krebs cycle. Acetyl-CoA comes from glucose. It also comes from fat breakdown. Acetyl-CoA is this 2-carbon molecule that goes into the Krebs cycle, and that's the 2-carbon engine that goes around and makes the NADH, again, that goes to the electron transport chain to make ATP. So it would make sense that increases in acetyl-CoA levels causes acetylation of histones, again, epigenetics, as specific lysine residues to loosen the interaction between negatively charged DNA and the histone, opening the chromatin structure to induce gene expression that promotes proliferation differentiation. What does all this say? It's what we said at the beginning, that epigenetics works by methylation and acetylation of our DNA strands to open up the strands to be available for the gene to be expressed, or to tighten up the strands for the gene to be hidden and not expressed. And this is just showing that acetyl-CoA, key metabolic player in the mitochondria, has an effect on what genes are expressed. That's all I'm trying to say here. Acetate is another one that causes this sort of expression. And acetate, as you recall, our microbiome produces short-chain fatty acids, one of them being butyrate and one of them being acetate. And acetate is another one of these that acts like acetyl-CoA to affect metabolism through epigenetic mechanisms. So the bugs that coexist with us also produce genetic changes within our own cells. Okay, amazing. And just to summarize, acetylation of enzymes or methylation diminishes their activity and slows down metabolism. We've stated this previously. This is feedback of excess intracellular acetyl-CoA. So just showing in this example, NAD, sirtuins, these are other molecules, basically all being players in the metabolism of acetyl-CoA to CoA, and some of the effects, the things that affect acetyl-CoA, and this is all linked to reading or hiding genes, which can be conserved through our offspring. So all I've been trying to say here today is that our genetics are not permanent. They are modifiable through nutrition and exercise, and these changes can be transmitted throughout our offspring. That's kind of how the theory of evolution works. Okay, so I will conclude this today, episode 6. You can visit us on miamivein.com, midtownbody.com. And what our team is trying to do, and this is my vein practice and the wellness practice, just trying to help folks fix their metabolic disorders through the poor standard American diet that we eat here in the United States. So we see a lot of obesity, diabetes, a lot of people on medications, driving chronic disease, such as heart disease, cancer, neurodegenerative disease like Alzheimer's. We've talked about this in the past. All this stuff is increasing, and much of it is probably reversible through a healthy diet and exercise. And all I'm trying to show you is some of the mechanisms as to why this stuff should start making sense. All right. Happy holidays, everyone. Signing off December 23rd, 2022. Miami Med GYM Podcast. Thanks for joining. Bye.